Dear students, my name is Ivana Šestak from University of Zagreb, Faculty of Agriculture, and within the module Smart Agriculture, I will talk about remote sensing and proximal sensing in agriculture. Firstly, I will introduce you with some theoretical foundations. In order to use remote sensing more easily, it is necessary to know the characteristics of a certain spectrum of electromagnetic radiation, as well as the composition and the state of the medium or object we are observing from the height. We need to have some knowledge about optics, but together with equal importance, we need to be very good experts in the field of the medium we are sensing. Is it soil or crop or rock? We need to have knowledge about soil science, agronomy, geology, hydrology, depending on the purpose of research. You can see here reflectance curves for three main natural medium. Natural covers like dry, bare soil, vegetation and water according to the visible and near-infrared spectrum of electromagnetic radiation. They totally differ between themselves, and these are typical reflectance curves. Agricultural areas on the Earth cover have a characteristic spectral reflectance, which includes vegetation, soil, and in some part water. Spectral reflectance of the vegetation curve manifests itself as absorbance and reflectance, and there are three types of energy interactions with plants, which we can see here on this graph. There is interaction with leaf pigments, so absorption in the blue and the red part of the spectrum according to the chlorophyll content. Then you see effect of the leaf cell structure, which causes increase in reflectance in near-infrared wavelengths. And then there is a moisture content, which can be seen in the short-wave infrared with typical absorption features. These are the dominant factors controlling leaf reflectance. Soil factors that affect soil reflectors are moisture content, soil texture, surface roughness, and organic matter. Later, I will explain in more detail effects of each of these factors. Reflectance of clear water is almost zero, approaching near-infrared spectrum. However, the spectral reflectance of water is significantly affected by dissolved and suspending organic and inorganic substances, as well as depth of water. So it is not the same if it is the sea or some marsh or a river. On picture down left, you can see different covers like clouds, snow, dry soil, moist soil, vegetation, clear water, turbid water. You can see differences in the visible and near-infrared spectrum. For example, here is the reflectance of the moist soil. This is MS abbreviation, so the reflectance is much lower than the reflectance of a dry soil. On the dry soil, some of water absorption features disappeared. Changes in spectral response in vegetation come from phenological changes during the season, beginning with germination, flowering, leaf development, aging, and stress. On the crops, absorption in the visible part of the spectrum is an indicator of the pigment in the leaves, from 0.4 to 1 micrometer, and the reflection in the visible and near-infrared spectra from 1 to 2.5 micrometers range contains different chemical groups that carry compounds such as starch, protein, lignin, sugar, and cellulose. Here we can see this picture upright. We can see effect on moisture in the vegetation, on vegetation reflectance curve. We can see the vegetation with less moisture has actually higher reflectance, and some of the absorption peaks regarding to the water content are less pronounced. They are still visible, but less pronounced. On the picture down left, we can also see standard deviation of reflectance with this bold line on vegetation according to the different chlorophyll content and regarding to the different color of the leaf. What we can see here from the wavelengths are visible and near-infrared. The highest difference between leaves and actually in the green part of the spectrum and in the red edge part of the spectrum, from red to near-infrared. Dark green leaf has a lot of chlorophyll in itself, but lower peak in the green. But leaves with less chlorophyll content are more yellow-green, so they have higher peak in the green reflectance. At near-infrared wavelengths, the variation of the spectral response within plant species is greater, and we can actually differ plants according to its cell structure. And this is visible here in the near-infrared wavelengths. For example, on this graph here, we can see differences in reflectance in near-infrared according to the different plant species. Plants have almost the same response within visible, while similar levels of pigmentation show differences in near-infrared because of different cell structures. And the reflected energy and the near-infrared refers to the crop biomass. What parameters can be predicted by spectral data? If we talk about the crop biomass, we can predict yield. There are direct predictions of total nitrogen content regarding chlorophyll because it is in correlation. Leaf area index as well, moisture content in plant, and then we can accordingly predict yield, biomass, nutrient deficiency. On some other wavelengths regarding cell structure, we can connect it to some other stresses like plant diseases or pests. Mostly used parameter in remote sensing of vegetation is spectral index or vegetation index. Vegetation indices are mathematical combinations of values of reflection at different wavelengths. Regarding the spectral curve for vegetation, we can compare and use visible and near-infrared wavelengths because the difference is according to the, as you see on this picture, healthy vegetation and vegetation under some stress. There are differences in the reflections of near-infrared and visible. We can compare it and put it into equation. Mathematical combination to get the index which has values from 0 to 1, and it's then easy to monitor vegetation according to the vegetation spectral indices. Here are the wavelengths which are used for calculation of NDVI. We can use it to monitor the density and health of vegetation, and we can estimate the potential yield during the growing season using NDVI, which can after that help us to optimize nitrogen fertilization. We can sensor and monitor NDVI during the early phases of the crops to calculate yield or optimal nitrogen top dressing fertilization, for example, regarding the generally vegetation cover on Earth. Some values can be connected to different land covers like less NDVI values, shrubs or grassland. Higher values are the agricultural production areas and forests. One parameter is also very important like spectral band. It is called the red edge from 670 to 760 nanometers, which is very important to monitor and to define the crop stress. Because when the crop is under any kind of stress like chlorosis, changes in chlorophyll concentration, changes in the cell structure because of some chronic stress like nutrient deficiency, pest diseases, red edge reflection moves to the shorter wavelengths. According to this change, we can find if there is any kind of stress. Here is the one investigation we established on our faculty when we monitor winter yield according to the spectral reflectance measurements during the growing season, during different stages of winter wheat, and according to the variable mineral nitrogen fertilization. We have achieved almost total correlation between grain yield and NDVI, which was captured during the stem extension phenophase. We could differ fertilization treatment groups according to the spectral reflectors and found that these specific differences happened in the green peak and in the red edge. So we use spectral reflectance as a function of the phenophase and nitrogen content in plant tissue. We estimated potential yield during the growing season using NDVI index as a base to optimize nitrogen fertilization. Vegetation indices are also used for soil survey. Here are some investigations which were done on soil using airborne and satellite data with different spatial resolution combined to predict soil organic carbon, which was very successfully. Here we will shortly mention satellite imaging and the power of Sentinel. From the Copernicus program, Sentinel-2 is the first optical Earth observation mission of its kind that includes 3 bands in the red edge, which is very different from all other satellites till now. The red edge provides key information on the state of vegetation. It is very useful in monitoring for agricultural purposes and agro-environmental monitoring. It has mission coverage and high revisit frequency of 5 days. It is cost-effective and we can use it for all kinds of agricultural purposes, for monitoring of crop conditions, soil survey after harvest, yield forecasting, adaptation to climate change, and it enables independent and very objective estimates of agricultural production. It has 30 spectral bands from visible near-infrared to short-wave infrared. They just differ in spatial resolution from 10 to 20 meters. Here is investigation which compares satellite Sentinel-2 and RapidEye. They are different in spatial resolution but also spectral resolution. One has 5 bands, Sentinel uses 11 bands and has low noise level compared to the RapidEye. Authors use satellite data to predict leaf area index and leaf chlorophyll. Sentinel-2 and RapidEye with spot 5 appear to be best. Sentinel best performed for leaf area and chlorophyll content according to its high signal to noise ratio. So very good and almost very high and excellent spectral quality. Regarding that, spectral bands and excellent geometry is more important than resolution. Now I will say something about proximal sensing and soil spectroscopy. Spectroscopy is a branch of physics that observes, qualitatively and quantitatively analyzes absorption or emission spectrum of specific wavelengths of electromagnetic radiation of a sample which is illuminated or excited by photons of different energies of electromagnetic radiation. Proximal spectroscopy is quantitative measurement of reflectance using portable devices. We go on the field and take measurements of the reflectance which can be used to build calibration models and prediction models of different properties of crop and soil, or we can use it also as a reference measurements for interpretation of UAV or satellite data radiation, which is used in proximal spectroscopy. It utilizes sun energy covering 0.3 to 2.5 micrometers, visible and near-infrared energy. Molecular bonds react with the energy. It can be found that spectral response of different material is determined by the reflection and absorption of radiation as a function of wavelength. Depending on the composition of the measured material, incident radiation causes vibration of individual molecular bonds via bending or stretching. The vibration absorbs radiation with specific energy quantum. We get spectral response. Shape of the absorption spectrum can be used for analytical analysis. Frequency at which the radiation is absorbed gives a reduced reflected signal, and we can register it in instrument as a percentage of reflectance. Wavelength at which absorption takes place depends on the chemical composition of the materials. Each material has its own specific spectral signature. We need optimal level of absorption. Some absorption has to happen so we can calculate and capture it. We use diffuse reflectance. So radiation penetrates in the material before reappearing, and then it can be measured. There is also effect of the particle size of different materials. Smaller particles have more scattering interfaces and photons are more easily scattered back to the sensor, but larger particles allow deeper penetration before scattering. We use proximal spectroscopy mostly in situ. Also, we can do it in the laboratory. But when we use it in situ, it should be done when the sun is high because it has effect on spectral response. On the picture on the right side, we can see spectral scans of mineral kaolinite. These graphs show influence of the incident angle of radiation on the possibility of detecting the characteristics of spectral curves of kaolinite. When the sun is higher, like on this picture, absorption curve is much more clear and more visible at 1400 nanometers. We can do measurements in the lab as well, while we use artificial light source, which is similar to sunlight on the Earth's surface. However, there is less reliable calibration and comparison of sample data with satellite sources if proximal measurements are employed in a lab. Regarding the soil, it is very complex and diverse media. You know that soil has two main constituents, mineral and organic fraction, and it is a mixture of numerous minerals at different particle sizes with humus and with different structure. When we take spectral measurements of soil, there is a high multicollinearity in the data. Through the spectrum from visible to near infrared, we can find overlapping and vibrational states creating several broad absorptions. And the reason for this is that most fundamental vibration of those materials occur in the mid-infrared, but it has overtones and combinations in both mid-infrared and near-infrared. In the visible part, where the radiation energy is higher than in the longer wavelengths, absorptions may occur through electronic transitions of atoms and also vibrational stretching and bending of molecules and crystals. Main factors that influence soil spectra are water content, clay minerals, organic matter, iron oxide content, soil texture and structure. Near-infrared spectrum contains useful information on organic and inorganic soil materials, mostly clay materials and soil organic matter. They have recognizable absorption features in this part of the spectrum. And water absorption belts are located here at 1400 and 1900 nanometers. As soil dries, they shrink. We can say that all these properties significantly affect spectral characteristics of soil. What parameters can be measured by proximal near spectroscopy? Total and organic carbon content and soil organic matter content, clay content, total nitrogen content, content of macroelements in soil, cation exchange capacity, soil moisture, soil pH, rare soil minerals, iron oxides and hydrocarbons as well. They all have specific peaks and absorption features in spectra. Soil moisture has a huge effect on soil reflectance. Soils with more moisture appear darker and soil reflectance is reduced. In the clay, soil absorption features of water still become visible even when the soil is dry. In sandy soil, absorption features disappear when the soil is dry. With increasing clay and silt content in the soil, moisture-induced absorption remain visible even during soil drying. This is relation mineralogy, water content. Clay and sandy soil texture is very connected to the water retention because it depends on the size of the soil particles. If other factors are kept constant, the reflectance increases as the particle size decreases and the surface becomes smoother. Iron oxide has wide absorption bands in the wavelengths of the visible part, 560, and then in the near infrared around 830 nanometers. Soil organic matter. Soils with higher soil organic matter content are darker. The color of the soil is strongly correlated with small amounts of soil organic matter. So soil can be black even with small amounts of organic matter of 5%, for example. But the rule is, as the content of the soil organic matter decreases, the color of the soil becomes lighter, so reflectance increases. With increasing soil organic matter is the opposite. Agroecological conditions in the field are very complex and numerous. Climatic areas and drainage conditions in the soil also have impact on the soil color and can have effect on the reflectance. We use reflectance for fine particle samples of the soil. Absorbance on this picture is logarithmic transformation of the reflectance usually used for larger particle size samples. Infrared wavelengths from 1000 to 2500 nanometers are used for a prediction of the soil texture and clay content. To distinguish certain clay minerals, we have secondary layered clay minerals like illite, smectite, and kaolinite, primary like quartz, feldspars, and sesquioxides or metal oxide like gibbsite, goethite, or hematite. We can use wavelengths in the infrared part to predict calcite, which is also specific absorption band from this part of the spectrum from 2300 nanometers approximately. And we can also monitor hydrocarbons because hydrocarbon acids are absorbed on clay particles and can be detected in this part of the spectra as well. Thank you for attention.